Hello, my name is Shelley, and my group B10 will be giving a lesson on the topic of GMOs. I will begin our lesson with an introduction to what GMOs are, which will be followed by Yorgo, who will give a case study with GMOs. Genetically modified organisms, abbreviated as GMOs, are defined by the World Health Organization as organisms such as plants, animals, or microorganisms in which the genetic material DNA has been altered in a way that does not occur naturally by mating and/or natural recombination. This organism can be modified through different technologies, including genetic engineering, which is defined as biotechnological methods used to manipulate an organism's genome. Humans have been manipulating the genomes of organisms for thousands of years, especially in the agricultural industry, such as through selective breeding. This figure shows two cows. One cow yields a lot of milk while the other cow yields low amounts of milk. As a farmer, in this case, would choose to select the breed of cow that yields a high quantity of milk in hopes of the offspring also yielding a lot of milk. Genetic modification is similar to selective breeding in the sense that both involve the manipulation of genetic material so desired traits are expressed. However, the two techniques are different, as selective breeding does not involve actively adding foreign genetic material into the genome of the target organism. Selective breeding involves breeding organisms that have the desired characteristics with each other to amplify the desired trait. With the use of biotechnological methods, scientists can artificially modify an organism's genome so the organism presents a new characteristic. This characteristic is advantageous and may include modifications such as resistance to pests or herbicides, drought resistance, resistance to certain diseases, or increasing the nutritional value and yield of a specific crop. In this video, we are going to examine how GMOs are produced through a specific example, the insertion of the gene CRY1AB into different crop species. This gene is commonly used in the production of GMOs. We will look at the advantages that having this gene gives to crop species and how the process of creating such GMOs is carried out. We will then look at what sort of crop varieties this gene has been inserted into and how prevalent they are in the U.S. Following the investigation of this case study, we will look more broadly at the public perception of GMOs and some of the associated issues with creating and selling GMO products.
A specific case study of the application of GMOs is that of corn plants, which are often eaten by the Ostrina nubilalis bug, known by its common name as the European corn borer. These corn borers bore into the corn stalk, making them difficult to get rid of using external insecticides alone. Moreover, the pesticides being used are becoming ineffective due to pest resistance. This situation creates the need for cost-effective, environmentally acceptable, and low-risk pest control tools for growers, such as BT protected plants. Scientists were able to find a solution to this problem through a gene called CRY1AB. This gene, found in Bacillus thuringiensis bacteria, encodes for an insect toxin called the BT toxin. This toxin is highly specific for moths and butterflies, and it was originally used to target the European corn borer. BT toxins have used a mixture of CRY proteins for the pesticides since the 1960s. The cloning and isolation of the CRY gene was done in 1981 in E. coli. The mechanism for producing GMO plants is as follows. First, the target gene to be inserted is extracted from the genome of the original organism using restriction enzymes. The gene is then copied and amplified using PCR and then inserted into a vector, frequently a bacterial plasmid. The plasmid is then inserted into a vector and put into a bacterial cell. The bacteria are then allowed to grow, amplifying the number of vectors with the gene. The plasmid amplified is inserted into a plant virus that can effectively penetrate into the desired plant cell through the cell wall. Different types of vectors, such as the yeast genome, can also be used based on the size of the gene that you want to insert. The viruses are then injected into developing plant cells in a petri dish or into a living plant. The desired gene will then insert itself into the plant genome, and these transgenic cells can be grown vegetatively. Another method of inserting a gene into an organism is shotgunning. In this case, the first three steps are the same, but a shotgun is used to insert the plant into the organism needed to be modified. Gold particles are coated with a plasmid to carry it through space from the shotgun to the plant. The gold particles are shot at the plant cells using a gene gun, allowing the vectors containing the gene to penetrate the plant cells. The plant cells now contain the gene and can be grown. Modified corn that contained the CRY1AB gene first became available on the market in 1996, with the specific targeting of the European corn borer pest. The gene was introduced in conjunction with other genes which allowed tolerance to certain types of herbicides. Since its initial uses in corn, the CRY1AB gene has been introduced into other crop species, including sugarcane, rice, and cotton. There are also variants of BT genes that are used to confer similar pest-resistant advantages. As of 2018, 82% of corn planted and 85% of cotton plants in the U.S. contain BT genes.
Genetically modified crops have been on the market since the 1990s, but were not required to be clearly labeled at the time, so consumers were not aware that they were consuming genetically modified organisms. Direct consumer surveys show that consumer knowledge of GMOs is very low and varies between countries. A 2004 study revealed that only 48% of consumers in the United States were aware that GMO foods were sold in supermarkets. Only 28% of Italian consumers reported that they had any knowledge of GMOs. This gap in knowledge about the science behind GMOs has resulted in some skepticism and aversion to genetically modified products. Consumer research shows that consumers in the United States are more likely to accept GM products than European consumers, but both groups are willing to pay more for non-GM products. This attitude difference makes sense when compared with the timeline of GMO history. GMOs were required to be labeled in Europe 16 years before they were required to be labeled in the United States. Despite studies highlighting the safety of GMOs currently on the market, there is still fear and misunderstanding surrounding GMOs. Many consumers worry that GMOs will make them or their children sick. A study published in 2015 compared opinions of the United States adult public and scientists from the American Association for the Advancement of Science. They found that 88% of scientists agreed that it was safe to eat genetically modified foods, but only 37% of adults did, a 51% difference. Furthermore, a comparative study that was conducted in China found that 46.7% of respondents claimed to have a negative view of GMOs, and yet only 11.7% believed that they understood the basic principles of GMOs. This perhaps highlights that much of the fear surrounding GMOs comes from a misunderstanding of how they function, and perhaps a concern that somehow the inserted genes may end up in their own body cells, causing adverse effects. The World Health Organization's International Program on Chemical Safety report on environmental health criteria for BT concluded that BT has not been documented to cause any adverse effects on human health when present in drinking water or food. Another study looked at the effect of the CRY1AB toxin on mammalian cells and found that it posed no acute toxicity.
Also, low scientific literacy may lead to misconceptions about reports on GMO studies, which in turn creates fear of GMOs. For example, a report was widely circulated that genetically modified wheat causes celiac disease. This is impossible because there is currently no approved genetically modified wheat on the commercial market. Additionally, a survey found that those who do not believe GMOs can be beneficial are also more likely to support the labeling of nanotechnology, a form of technology which improves the material's function by altering it at a micro scale, perhaps indicating that views on GMOs are tied to a general sense of distrust in scientists. Additionally, some concerns over the use of GMOs do exist within the scientific community and are often the basis for political opposition. One such concern is that GMO crops could crossbreed with wild populations and introduce the modified genotype into the wild in an uncontrolled manner. It is argued that making sure that cross-pollination does not occur is difficult to manage, and the effects of introducing the modified genome into the environment is not well studied. Furthermore, there is a concern that the proteins produced as a result of the introduced gene may be an allergen or toxic for some individuals. Additionally, there are concerns that the overuse of GMOs may create monocultures, which have limited variation and therefore may be more susceptible to destruction from a new pest or disease. Overall, it becomes clear that GMOs have the potential to confer significant advantages regarding food production, but they must be managed and studied very closely in a way that is transparent for the public.